I'm going to dive right in, and we'll have plenty of time for discussion. And I think this should be a fun presentation. We love this material a lot. And the presentation we're giving is based on a long article that's just been published in the Michigan State University Press book, Darwinism, Design, and Public Education. And it's the result of, as it happens, 5 years of research. So we hope you enjoy this, and we'll point you to that resource if you want to get more information than we're able to cover in our presentation here. The Time magazine in 1995 had a cover story on what they called evolution's Big Bang. And this was predicated by a recent discovery in China of some extraordinary fossils that showed the Cambrian era animals in exquisite detail. The preservation quality of these fossils was just incredible. And these fossils were confirmed and provided more information about an event that scientists have called the Cambrian Explosion. And they were found in southern China in a province called Chengjiang. Marcus has been there for a conference on this material in the year 2000, and it was 1999. And this is the site here with the you see the Chinese markers here distinguishing the Cambrian and Precambrian strata layers. And of course, the Chengjiang was not the only big Cambrian find in the world. The other really famous find is the Burgess Shale. But the Chinese were quite proud, and arguably so, at the preservation quality of these fossils and the additional information that they provided about the Cambrian, which will be part of the basis of our talk today, because in essence they showed that the Cambrian explosion was more explosive, more sudden, broader in extent than had previously been known, and many new representatives of new phyla have been discovered as a result of these Cambrian fossils, new organisms, things like fishes that we knew about before that we now know originated earlier and more suddenly. So we'll look at some of these data along the way, and just right now I want to orient you. The Cambrian explosion takes place in about on the standard geological timescale at 530 million years ago, and ends now they're thinking within 5 million years, maybe a little more, maybe a little less. 5 million to 530 to 525 is the increasingly accepted time frame for the Cambrian explosion. Well, what exactly is the Cambrian explosion? Let's just orient ourselves a little bit more here. The Cambrian explosion refers to the geologically sudden appearance of many, in fact, most of the major animal groups, what are called the animals as represented by disparate or separate body plans. And if you think of a body plan, if you've heard that term tossed around, a way to think of a body plan is as a distinctive set and arrangements of body parts or organs and tissues. And on the slide here we have 3 different examples of body plans, separate animal body plans. We have the chordates and the arthropods and the cnidarians, just 3 examples. And you see how different are the different ways of organizing body parts here. The chordates have their dorsal nerve cord, their internal skeleton. The arthropods are segmented, and they have an external skeleton just for starts. And of course, the cnidarians, as represented by this beautiful jellyfish, have their own distinctive mode of organization. Now, when paleontologists and taxonomists talk about body plans, they're usually talking about differences that are at the phyla or class, phyla, subphyla, or possibly class level in the biological hierarchy. We're talking about the origin of the major divisions in the animal kingdom, the major separate divisions. So not the species or genera, but rather the phyla, subphyla, or classes. And the Cambrian explosion is marked by the sudden appearance of completely novel body plans, that is, completely novel phyla coming into the fossil record. And So what we want to do now in our talk, and we have a little more time than the other sessions. We've got the full hour and a half, and we're going to kind of pass back and forth different parts of this, talk to each other, and leave plenty of time for discussion as well. We want to do three things. We want to talk about the Cambrian explosion, and in particular, we want to address three questions. Why is this so extraordinary? Why is the Cambrian explosion so extraordinary? And Marcus is going to address that by describing the key features, the descriptive features of the Cambrian explosion event in the fossil record. The second thing we're going to address is why are scientists so perplexed by that? And in particular, we mean the orthodox neo-Darwinian scientists, because as you'll see, the data from the Cambrian contradict Darwinian expectations and predictions at every turn. And I wanted to say almost, but really it's let's say virtually every turn. This is really a tremendous challenge to the Darwinian theoretical framework. And given that, we want to then in the end talk about how we might better explain the data from the Cambrian explosion within a design theoretic framework. That is, we want to offer intelligent design as an explanation for the Cambrian and its key features, and explain the rationale, that is, explain why design should be considered as not only an explanation, but we're going to argue as the better and best explanation for the key features of the Cambrian explosion. So we're going to kind of divide the presentation in those three parts. Marcus is going to start by talking about the distinctive features of the Cambrian. Marcus is a paleontologist. He's already earned a master's degree from South Dakota School of Mines, and he is finishing a doctorate now from the University of Rhode Island. Did I get that right? I've once said Delaware, and I meant Rhode Island. So he's an up-and-coming talent in the design movement, and we're really happy to feature him. He's done some really terrific research in contributing to this multiple-authored paper that I mentioned at the beginning in the Darwinism, Design, and Public Education book. The paper was authored by Marcus and me and our colleagues Paul Nelson and Paul Chen, who is a marine paleobiologist at the University of San Francisco. So Marcus, go ahead and take over.
Thanks very much. So, the question is about the Cambrian explosion. Why is this so extraordinary? Why is it so perplexing to standard Darwinian evolutionary biologists, neo-Darwinians and punctuated equilibrium proponents, such as the people that I study with each and every single day of my academic career? Well, one of the major reasons is that we have this first appearance of most of the animal body plans, and we'll see exactly how many that we're dealing with as we go further on through the presentation. So, as Stephen mentioned, when we're talking about major animal body groups or body plans, or bowel plane is something that Stephen Gould had referred to these things as, we're talking about basically here the phyla level. And here we have representative groups of the phyla, such as the Brachiopoda. These are the lamp shells. They look kind of like clam shells, although they're a fundamentally different organism in terms of their biology. They both have shells. That's about where the similarities end. This curious organism next to it, Eldonioida, not exactly sure even what this thing is. It appears to be a worm that's converged upon a jellyfish morphology. It's got this large frond-like sort of covering. The two things straggling off to the side happen to be little attached brachiopods that are suctioning onto it. But on the internal section of Eldonioida, you actually have a worm. So this thing is not a jellyfish, it's a floating worm of some kind. And we have various other things that you might be a little bit more familiar with, particularly the arthropods and the chordates, represented by a little fish and a trilobite. So, as I just mentioned about the Brachiopoda, it's a basic plan for this thing. It has a shell of two unequal parts. If you look at a clam shell, you open it up, you've got a mirror image of the two shells, one next to the other. Well, the brachiopod is different in that the two shells aren't the same. But if you look down on the top of them, they're a mirror image. And so rather than opening up a clamshell and seeing the mirror image, what you do is you look down on the brachiopod, and both sides look the same. If you look at the other shell from underneath, both ends look the same, but it's not the same as the top shell. I hope that kind of helps. The feeding organ for the brachiopod is very different. It's this big spiral filter feeding sort of thing called a lophophore. And we see those in a couple other organisms, not mollusks, not bivalve shells. The phylum Hyolitha, not exactly sure what this is. It seems to be closely nested to the mollusks, although it has entirely different musculature. Internal skeleton or internal musculature is different. The outside shell is different, and it has this interesting little attachment lid that it's able to kind of pop open and close in order to feed and protect itself. Some of the other phyla that we're dealing with are a group of worms called phoronids. They, like the brachiopods, have this lophophore feeding organ, this spiral filter feeding sort of thing. And they tend to burrow and form a secreted tube around themselves, so they pop up out of the ground or out of the ocean substrate, feed for a while, and then pull back in at any sign of danger. Now, we're still going through all different types of phyla here, and one of the curious things about phyla is we got a lot of worms. Dogs that have worms, that's a different phyla. These are worms, these are different phyla. The priapulid worms are a carnivorous group of worms. The proboscis that's located in this circle here will go down and grab onto something and start chewing into it. It's got little tiny hook-like teeth at the end of this sort of thing. And it's got a straight digestive tract. Annelids are slightly different. They're also segmented worms like the priapulids, but they tend to have bristles for locomotion and anchoring. They have these little fibers all along the side. So the earthworms that you have in leeches, these are the two modern representatives that we're most familiar with, hopefully not as familiar with the leeches as we are with the earthworms. Tardipolypoda, this was a new phylum that Chinese paleontologist Chengjiang China had erected. We'll see exactly whether or not this thing survives in the standard literature or not. But for the moment, this is a group of extinct worm-like segmented things. They're not exactly arthropods. Even though they have these little appendages, they appear to be some kind of tube foot that's soft-bodied and not a hardened exoskeleton with internal musculature such as the arthropods have. This is a fundamentally different sort of thing, and they don't have antennae, and it's questionable what kind of eyes they happen to have. And we have these as fossil representatives. On the right-hand side is one of these fossils that has come out of China. And the thing to remember here with these worms that I've shown you and this tardipolypod organism, these things are very soft-bodied. These things don't tend to have hard parts. And so what you're looking at is a real biological residue that's been left in the fossil record. This stuff, when you collect it out of the rock, is like smudge. You can just drag your finger over and wreck the fossil because it's preserved in a very, very fine clay. But because of this, we're able to preserve such excellent detail on these things. It's positively exquisite. You can see the gut cavities on these sorts of things. And in the dark middle section, as you go further from the head up by the two, and you go further down, you see a dark line in the middle. That's the gut. Now, as a paleontologist who tends to work with vertebrate animals and not these little spineless things, in more ways than one, to just give a little bias here, I tend to work with skeletons. I tend to work with teeth. My study is in mosasaurs. These are big swimming reptiles. And I never get stuff like this. You go to the Niobrara Chalk in Kansas, which is the most proliferate vertebrate deposit probably in the world, and you're still just getting skeletons. You're not getting soft parts. And I can't stress to you enough how rare it is to find this type of material. You have this place in China, you have Burgess Shale, you have a few other places around the world, some others in China with dinosaurs and the like that have biological residue left here. But we're getting an extraordinarily unique glimpse into the biological system that was in existence at the Cambrian that we very rarely get to see. Some more weird stuff. Problematica here. This is a fossil called Deinamiscus, and it appears to be some sort of stalked animal. I mean, this isn't a plant of any kind, but it has these feathery rods that stick out, which are probably some kind of food collection organ. But this thing is very poorly understood. It's only known from Burgess and from Xinjiang. Really don't know exactly what this is. And so the phylum Problematica is not a real phylum. That means it's problematic, and we don't really know where it goes. But it is fundamentally unique in its body organization, and so we'll see what happens with the taxonomy of this thing. As I mentioned, we had this weird thing called Eldoinioida. Once again, this phylum level, don't really know. I mean, this thing is a worm encased in a jellyfish, kind of. You can note the little kind of frond things sticking out of the bottom. This is a food collecting organ, and it has a couple little critters attached to it that are either hitched for the ride, or one of the other possibilities is that these things were predating them or scavenging them once they had hit the ground. Another floating organism is the ctenophores, and these are different from jellyfish. They actually have a a body symmetry that is based on 4s and 8s and numbers like that rather than truly radial. And each one of these little number 2s here represents a series of filaments that all beat in synchronized form in order to move these things through the water column. But once again, it's kind of like a jellyfish, but very, very different. It's kind of, you might think, functionally similar to a jellyfish, although biochemically and in terms of body composition, radically different, which is why it's in a different phylum altogether. Some things that might be a little more familiar to them, besides, say, the annelid worms, are the phylum Arthropoda. And we're going to break up the arthropods because not only do we have arthropods in the Cambrian at this particular time in Earth history, we have a lot of different types. And so we're not only seeing a completeness in terms of the broad range of various different animal phyla, but in things like the arthropods, we're seeing major groups of arthropods at the subphylum or the class level that are also making their first appearance. So we're seeing a magnificent breadth of morphological disparity here amongst the various groups. Here we have the subphylum Crustacea. Our favorite little edible types are shrimp and lobsters and crabs, and this is a more primitive form called Waptia. It has kind of this bivalve sort of shell over the top of it. It is segmented like all other arthropods. It has an exoskeleton, although the exoskeleton on this is probably a non-mineralized type. You squish a bug and it crunches because the keratin is mineralized. It has some extra material in it that makes it hard. Some of these crustaceans have an exoskeleton that is not particularly well mineralized, and so it's more of a tough outer covering rather than a hard, crunchy kind of shell. By the way, I'm not particularly sure whether or not these guys would be really tasty or not. We don't get that kind of opportunity in the fossil record. It tastes like mud. So some of the subphyla that we have here are the crustaceans with things like Waptia. Trilobites are their own subphylum. They're a radically different group within Arthropoda compared to the more familiar stuff that we know of insects and crabs and these sorts of things. Trilobites are very much a unique group that has its own morphological completeness to it and disparate position with respect to others. Another group that's fairly unique is the Anomalcarida, and these were kind of the lions of the day back in the Cambrian. These things were pretty big as far as things go in that time, and by saying pretty big, I'm talking this big, to this big. Now, that's not particularly huge by our standards. These things could get a meter or so in length, but if you're a trilobite who happens to be about 4 centimeters long, you start sweating when you see something like Anomalcaris, which seem to be designed very, very nicely to pick up trilobites with these front grabbing arms, drag them internal to a mouth that is located on the underside of the body, crunch into the backside of a trilobite, and suck out the guts. And we see trilobites in the fossil record that have a unique pattern of bite marks on their hindquarters. And what we've realized subsequently to finding more complete versions of Anomalcarida in places like Chengjiang and the Burgess Shale is that we're seeing a predator that preferentially attacks the hind end of a trilobite by grabbing it, dragging it to its mouth, and biting off the back end rather than the front. Apparently it was easier. And so we're seeing some, in addition to all this phyletic completeness, we also get a good look every once in a while at predator-prey relationships within the fossil record, just to go off on a bit of a tangent. So we're still in arthropods. I mean, these things are tremendously diverse, and there's major, major differences between these sorts of things. The body plans include, for the Anomalcarida, these giant appendages in the front, and they have a round mouth. If you think of, science fiction movies where there's this portal that locks open and locks closed like a big circle, and you've got, a bunch of teeth that kind of all come together, that's the sort of mouth that Anomalcarida has, and that's kind of displayed here with the number 2 that you can see, the round mouth there. Now, the part that everybody seems to be interested in, chordates. Now, in reality, chordates make up a relatively minor portion of the fossil record as a whole, and not particularly a whole lot of the fossil record in Chengjiang. But we tend to have what I might call a little phyletic bias when we're looking into chordates and the like. We're chordates, we want to figure out what it is that's us in the fossil record. And so we look at the chordates, and there's actually 3 different subphyla of chordates in the world, and we have basically the vertebrates or the craniates, more specifically to include the hagfish and the lamprey, which aren't technically vertebrates. We also have basically cephalochordates, which look kind of like chordate larvae. And then we have tunicates, the sea squirts, that are radically different from those as well. So the basic body plan that we look at with a chordate, for example, is the dorsal nerve cord. That means the nerve cord is on the back side of the organism. In arthropods and some other types of worms, for example, it's on the ventral side. It's on the chest side, so to speak, of the animal's body. Vertebrates have a backbone. Craniates, to which vertebrates are part of, have a stiffened notochord, and within vertebrata, we also include a backbone with that. So here we have a representative sample. This is an organism called Hycoichthyes, and I won't even try with the last name, don't worry about that. Hycoichthyes is a small hagfish-like chordate, and it had a dorsal fin, which is represented by the line with the dashes that kind of go back into the rock. As I said, these things are very, very delicate fossils, and this is considered beautiful. Just to give you an idea of what we're looking at here. The detail is very interesting. You can almost see some of the ribbing down on the ventral portion, the bottom portion of the animal. Those are muscles, or that's the fossil remains of V-shaped myomeres. These are the types of muscles that fish have when you have a nice piece of cod or what have you, salmon and catfish. The flaky type of stuff that you eat, that's the type of musculature that we're seeing in fossils that are determined by standard geology to be 530 million years old. So the preservation here is simply unbelievable, and we might even have evidence for eyes and the like in these types of organisms. So we have a problem. It is both extraordinary and perplexing. We have the first appearance of most of these animal body plans coming in a geological millisecond sort of situation. So here I tag my partner back into the ring and bring back Steve Meyer.
All right, there was several other features of the Cambrian that we want to address. We've already talked about the idea of sudden appearance but I want to amplify that. The key feature we call sudden appearance without evidence of ancestors, without evidence of transitional intermediates. And if we get a sense of the time scale here, if we're in the present, the Precambrian begins the Cambrian begins 543 million years ago. The Cambrian explosion takes place within that Cambrian period, starting at about 530 million years ago. The date for the Cambrian has been redated about 10 years ago. People used to refer to it as an explosion when it was more on the order of 40 to 70 million years in extent. And as a result of some zircon crystals that a zircon dating method and some other type sections in Russia that extend into China, it's been possible to really nail this down and realize that the explosion itself is a 5 to 10 million year event at the outset. J.Y. Chen in China and some of the paleontologists that are working on the evidence in China will privately tell you they think it's more like 2 to 3 million years or maybe less. I mean, this is geologically instantaneous. It's very extraordinary. And one way to get a handle on that is if you take the date of a 5 million year explosion 530 million years ago, and you think of the time scale of the Earth starting at 4.5 billion years ago, the idea here is that we have an event that takes place, and you think of the history of the Earth as a football field from origin to present. The Cambrian explosion takes place on the defensive team's 12-yard line, and it represents about 4 inches of the football field that represents the history of the Earth. And yet you have, as Marcus showed, and we'll give you the exact numbers on this, a majority of the major animal forms of life that have ever existed come suddenly into the record at that point. So this is quite extraordinary, and we'll see unexpected. Here's a slide I just love from a teaching standpoint, because if you think of the geological law of superposition, which is basically the idea that generally speaking, the sediments on the bottom are going to be older than the sediments on the top, you see that the Cambrian represents a sudden appearance of all these different and disparate forms of life in a very narrow seam of rock essentially all the way around the world. We found there are many Cambrian finds in addition to Burgess and Chengjiang, but so we've represented this with a rock column on one side and the geological scale with the time numbers on the other in parallel, and then you have the animal forms. In reality, I was talking to Marcus yesterday, the Chinese units are about, that represent the Cambrian explosion, are about 30 feet of rock. And you have, I was an oil company geophysicist, and we dealt with sediments from the Pleistocene in the Gulf of Mexico, an unusually thick unit, but just that one little geological period had over 9,000 feet of sediment. So this is a very narrow punctuation, a sudden appearance, geologically speaking. The third feature of the Cambrian that goes with this, that is a corollary to this, is that not only do we have a sudden appearance of new form, but we have a sudden appearance of new information. Because to build biological form, you need information, and as we'll see, you need that in the form of the digitally encoded information in the genome, but you need that in there are epigenetic, other sources of information necessary to build a body plan. And so if we think of a new animal, we have to think about at least the new code and the other forms of information necessary to build that animal. I often ask my students, if you need to give your computer a new function, what do you need to provide it? And given that they're better at computer games and things like that than I am, they immediately get it that it's what you need for new function is new code, new information. It's the same in biology. New function requires new form requires new information. So when we see these marvelous new forms appearing suddenly in the fossil record, we understand that a necessary condition of the appearance of that form is new information, both genetic and epigenetic. And then one way to get a handle on that, to quantify that, is to think about the number of cell types represented by these new phyla that arise. The sponges, which are a phylum that exist in the Precambrian, have on the order of 5 cell types or so. So that when we get, and cnidarians are also a Precambrian phyla that exists. There are a couple of more primitive phyla that exist in the Precambrian. But when you get to the Cambrian, you get from a jump of, the Precambrian phyla are on the order of 5 to 10 cell types. With arthropods and chordates, you're talking about 50 or 60 cell types. Now, every cell type requires dedicated proteins to perform the distinctive function of that cell type. If you're a cell in the gut and you are involved in digestion, there are specific digestive enzymes that are specific to that cell type. And so therefore, a new cell type necessitates, implies the existence of new proteins, which in turn requires at least a lot more genetic information to build those proteins. So this is maybe a somewhat crude way to get a handle on quantifying the amount of information, but if you recognize that there's a big jump in the complexity of these organisms as measured by the new cell types required to build the phyla in question, then you realize that you also are talking about at least a massive increase in the complement of genetic information required to build those cell types. So one way to get a handle on that quantitatively, this again is rough, but you think of a single cell at about 400,000 base pairs, but an arthropod, modern arthropods have about 120 million base pairs in the coding regions of the genomes of these different types of organisms. So this is gives a sense of the kinds of jumps we're talking about in complexity as measured by genetic information. Precambrian sponge, 5 cell types. Cambrian fish, 60 cell types. That represents a lot of new information. A further feature is what we call persistent morphological isolation. You may be quite familiar with the Stephen Jay Gould, Niles Eldredge model of punctuated equilibrium. They call this stasis. I'm not quite sure why we came up with this term, but it seemed to help because it really explained what we're talking about, at least if we define each term. Morphology refers to body parts and their arrangement, the arrangement of form, the distinctive forms that we see. Isolation is the idea that you have major differences in form between the phyla, between the new animals that arise in the Cambrian. And persistent means that these major differences stay constant, unbridged by intermediates throughout the history of life. Marcus showed a picture of a beautiful comb jelly. And yeah, here we have it. It's just absolutely extraordinary. The picture down below is an artist's rendition of the fossil find of a ctenophore, a comb jelly, from the Chengjiang Formation, the Maoshan Formation in Chengjiang, China. The little picture above it is a picture of a modern comb jelly from the Monterey Aquarium. And if you look at these things closely, you realize there has been no drift, no evolution, no change in the basic form of these comb jellies over 530 million years of geological history. So this is an extraordinary stasis, a lack of directional change, and moreover, and this is the part I think that we're wanting to emphasize as opposed to just the concept of stasis, the comb jellies have remained the same in their basic form. The basic body plan of the chordate, the vertebrate fishes, also have remained very constant through time, and the difference, the disparity between these two different forms has remained unbridged through geological time. So we're talking about a picture of the history of life taken on its face. What we have here, I think, is strong evidence for some kind of what's called polyphyletic typology, the idea that you have major separate groups of organisms that arise independently and remain separate from each other over time, rather than the classical Darwinian picture of the monophyletic, the single tree of life picture of the history of life. But we'll get into that more later. And one way to illustrate this is to think about our own technological innovations. Automobiles have a distinctive arrangement of parts, a distinctive body plan. Airplanes do. And the space between them is unoccupied. There's a humorous cartoon in the book, What's Darwin Got to Do with It, that shows a car plane or a plane a car. And it's really obvious from the cartoonist's drawing that this thing won't fly, nor is it going to be very effective on the I-5. You know, it's an intermediate form morphologically that just doesn't seem to do either of the two functions well. And so there's a kind of functional logic here that when you have very disparate things that have distinct functions, the combination, the combination form, the morphed form, is not going to be very effective. Maybe that's why they're not found in the fossil record. In any case, they're not, and this gives a little handle on this. So this is a striking feature of the fossil record that we think needs explanation. Somewhat and closely related to this is what's called the top-down pattern of appearance in the fossil record. And what's meant by this in the strictest terms is simply that the representatives of the disparate higher taxa appear first, only later to have a multiplication of other representatives of these same taxa. So the higher taxa, the phyla and classes appear first, represented by one or very few members. And then later there's a multiplication of other members of that same larger group. So you have, the theme comes first, the variation on the theme is elaborated later, okay? And this is called the top-down pattern of appearance. And this is one of the really puzzling and perplexing things about the Cambrian explosion, as we'll see later when we begin to talk about the Darwinian prediction. So you can think of, you know, these major differences that Marcus has elaborated here so nicely between, say, arthropods and fishes. And initially you have only these very few representatives of the chordates, but then later you have a diversification, and you have many, many, many different varieties at the species, genus, family level arising later. So the way the paleontologists say this is the disparity, the big differences appear first, the little differences, diversity, appear later. So disparity before diversity, or top-down, the top-down pattern. Now, so we have 5 main features of the Cambrian explosion, and any good theory of biological history, of the origin of the animal groups, is going to have to account for these features. And so we're going to look for some answers here, and of course we're going to look first at what classical Darwinism and the updated neo-Darwinian theory would predict about the fossil record, or we might also talk about just simply empirical expectations. We are dealing with an historical science here, and the kinds of predictions that historical science generates are more of the form what we should expect the evidence to look like when we find it, rather than something that you can produce in a controlled setting in a laboratory. In any case, common descent, which is one of the two main parts of Darwin's theory and of modern neo-Darwinism, envisions that the history of life should look like a single branching tree. Now, this is results from Darwin's understanding and classical neo-Darwinist understanding of the way in which the mechanism of selection and variation works. The idea is that you have many small incremental changes, genetic changes, that confer, when things go right, a functional advantage, and when those functional advantages accumulate over time, eventually you produce significant differences. But the mechanism adds up little tiny genetic changes bit by bit by bit over time. And so what you then expect for your picture of the history of life is a continuous tree-like picture where you start with something very simple, maybe a single-celled organism of some kind, and then eventually you get more complex single-celled organisms, then multicellularity appears, and then the diversification events. So the tree of life picture is part and parcel of the Darwinian picture of the history of life, and it is generated, it's a theoretical implication of Darwin's own understanding of the creative power of natural selection acting on random variations. But the idea that natural selection is creative, but only very slowly and gradually and incrementally. It's the small, in neo-Darwinian terms, genetic mutational changes that get added up and over a long period of time. So the first prediction, or empirical expectation, to be more precise, of neo-Darwinism is that there should be a gradual increase of biological complexity, and with that gradual increase, the existence in the fossil record of numerous transitional forms leading to the big differences, to the phyla-level body plans. So you should start with little tiny differences, and they should add up. And so this is the classic tree picture of the history of life. Then we have the time axis in the vertical, the morphology, differences in form in the horizontal, and differences in form should emerge very gradually, but then eventually add up to become very great over great expanses of geologic time. The Cambrian is a challenge to this, because as we look at the fossil data at face value, at the level of the body plans, we don't see all of these smaller changes gradually adding up to produce those big changes. Instead, we see the big changes suddenly, and many of those big changes persisting over time, some going extinct, a few arising before the Cambrian. There are 2 or 3 phyla that exist in the pre-Cambrian strata, but even those arise and then keep going virtually unchanged. So you have, instead of the tree-like picture, you have something more like a series of parallel lines, or to be more precise, a series of something more like an orchard with a series of trees that do have some diversity and variability, but that have independent beginnings and stay separate from each other over geologic time. So this is the contrast between the Darwinian theoretical expectations and what the fossil record actually shows when we're talking about the origin of the phyla and subphyla. Here's another picture that we've stolen from our colleague Jonathan Wells that I think makes this really nicely clear, as when we look at the contrast between what the theory would predict and what the evidence actually shows. And another slide that makes that clear. So Darwin was, of course, aware of this, though at the time there were many fewer phyla to be reckoned with. But he said in the origin, he said the case at present, speaking of the Cambrian, the case at present must remain inexplicable and may be truly urged as a valid argument against the views here entertained. So this is not even a new challenge to Darwinism and neo-Darwinism. It's been around for a long time. It has just become more acute with the discovery of more morphological disparity, more different phyla, each of which are lacking these transitional forms, the ancestral precursors that would be necessary to fill in the Darwinian typology, the tree of life picture of the history of life. And modern neo-Darwinists are aware, acutely aware of this problem as well. Even Richard Dawkins, who is not prone to making concessions in argument, says it is though they, the animal phyla, were just planted there without any evolutionary history. So you have the appearance of sudden appearance. The second prediction that the Darwinian model makes is what you might call a bottom-up pattern of appearance. And this is really, in a sense, a corollary to what we've just been talking about. The idea is that diversity at the species level, at the genus level, species and genus level, should appear before disparity at the higher taxonomic levels in the phylogenetic scale. So here's Darwin himself spelled this out. He said, because of the way the mechanism works, where natural selection is adding up little tiny changes bit by bit, you should first have varieties within a species, and then there should be divergence, and a new species should emerge from separate varieties, and then new genera should emerge, and then finally the changes would be big enough that we would classify those differences as new families and orders and classes and so on. So if you think of the body plans and the phyla as big morphological differences, those aren't going to emerge until after a very long period of evolutionary change. You've got little tiny changes. We're going to classify those little tiny changes as varieties within species. Little bigger changes are going to be separate species. Little bigger changes still, different genera, and so on. So the big differences in form correspond to the higher levels in the taxonomic category. Since it takes a long time to produce big differences in form, it's going to be it's going to take a long time before we generate, on the Darwinian model, new phyla. And so we can kind of animate this and think of starting with one species and then going to having enough change that now we have at least a genus with several species, and then more change. Now we can say that we've got several genera, and then eventually families, and on up the scale. So it's going to take time for the branching tree to branch out. And so on the Darwinian model, you should have diversity, the small changes first, and disparity, the large changes, later. And it turns out that even the much ballyhooed and vaunted punctuated equilibrium model makes the same prediction. It's just that the jumps are going to be a little more discrete and sudden rather than continuous and gradual, but you still build to the phylum level differences from the bottom up. Okay, in the punctuated equilibrium model, the mechanism is understood to be something called species selection. Natural selection, instead of selecting for small changes within a species, it selects for, in the thinking of the Punk-Eek people, it selects for the most species among a group of species. So the unit of biological change that natural selection selects for is bigger, but still you start from the bottom and build up from species to finally to phyla level differences. So it's the same basic picture bottom up. Well, of course, the problem that is, as we've already noted, that the fossil record manifests exactly the opposite pattern. Here, Roger Lewin, the science writer for Science magazine, back in 1988, said that there are several possible patterns exist for the establishment of higher taxa. The two most obvious are the bottom up and the top down. In the first, the evolutionary novelties emerge bit by bit, but the Cambrian explosion appears to conform to the second pattern, the top-down effect. So we have the big differences appearing first, with the more minor variations arising subsequent to that in later periods, later geological epochs and on the time scale. So you have the Cambrian explosion with the big differences appearing, the smaller differences appearing later. The theme comes first, the variation later, precisely the opposite of the Darwinian picture. So here's a way of getting a handle on that with a nice graphic. You have the big difference between the arthropods on one side of the slide and the vertebrates on the other, and the vertebrates within chordates, and this is the fish. And then later you have the diversification of those vertebrate fishes, but the big difference is there from the beginning and is never bridged. So here's Irwin and Valentine. They say most higher taxa were built from the top down rather than from the bottom up. The fossil record suggests that the major pulse of diversification of phyla occurs before that of classes, classes before that of orders, orders before that of families, and so on. Exactly the opposite of the neo-Darwinian prediction. Now, there's a third prediction that Darwinism makes, the idea that the morphological distance between the organisms and thus the number of phyla will increase gradually over time. And I'm going to let Marcus explain this, but I also want to tout a little bit some of the research that he's done that have enabled us to nail this down. If you've read popular books about the Cambrian explosion, you will sometimes hear the claim that all of the phyla arise in the Cambrian. There are other people that dispute that utterly and want to, say that many phyla, lots and lots of phyla emerged earlier than the Cambrian, so the Cambrian explosion is not that big a deal. And as often happens, the truth lies in between those two extremes. Turns out Gould is incorrect about this. It's a popular claim about his claim that all of the phyla emerge. Marcus has actually done a really wonderful piece of research, an exhaustive literature search, to give us some quantitative data on just how many phyla appear and what percentage of the phyla appear in the Cambrian. On the Darwinian model, we would expect the number of phyla to be kind of clicking along, gradually increasing over time. So you'd have a very smooth, if you're just kind of keeping track of phyla, click, click, click every time a new one comes online, you would expect your numbers to be increasing gradually and smoothly over time as those differences in form are gradually adding up to be big differences. What's striking about the Cambrian is you get this spike in the number of phyla. Marcus is going to tell us how big the spike is and what it represents as a percentage of the total number. This is some original research, and it's one of the really nice features of the paper that we've done. Go ahead.
Thank you, Steve. Apparently, we're either going to have to open the double doors to get my head out the door, or perhaps deflate some stuff here. So, all right. As Steve had just mentioned, the standard prediction of Darwinian evolution, neo-Darwinianism, punctuated equilibrium, pretty much any major theory of universal common ancestry is going to assume that over time, more and more phyla are going to be added. Now, the time amount here that I'm talking about could be arbitrary. It could be millions and millions of years. It could be several million years. But what we should see as a signal in the fossil record over time, going, say, from somewhere in the Precambrian up through the Cambrian into the Ordovician and Silurian, is that generally you're going to start off with one phylum. I mean, all the animals, according to standard neo-Darwinian and Punk-Eek evolutionary systems, assume that all the phyla descend from a single common ancestor. And so at one point in time, there's going to be just one phylum. Now, this is a simple doubling pattern here that just took this as kind of a null hypothesis that we will have doubling branches, that one phylum will give rise to two phyla. Those two phyla will eventually split, and there will be four phyla, and so on and so forth. And so that's, the time here is arbitrary. I'm not trying to pin an evolutionist down and say that it has to happen over a certain amount of time in particular, but over time, this should be a pattern that should be coming out of the fossil data if their prediction is correct. So we have to ask ourselves, when we first start seeing animal phyla, what does this look like? Well, in the Precambrian, in a section called the Vendian or the Neo-Proterozoic, to get a little specific, we have basically somewhere around the order of 3 animal phyla. We have sponges, we probably have cnidarians, and we probably have some kind of worm that's making tracks and trails that we recognize in the fossil record. Now, there's the potential that we might have one or two others. It depends on highly speculative interpretations of often some very bad fossils. And so we're trying to be as generous, yet at the same time we also want to be realistic. So we're going to say in the Precambrian for the moment that we actually have hard evidence in terms of body fossils and trackways that indicate that there are at least, but probably not much more than, 3 animal phyla. As we've gone through and I've talked about some of the fossils that we've seen in the Cambrian explosion, I've hit a few of the phyla, the chordates, the arthropods, preapulid and annelid worms and things like that. But I didn't hit all of them. We actually have 19 animal phyla that make their very first appearance in the Cambrian, directly above some of this Precambrian material that is also fossiliferous and giving us the animals in the Precambrian, the 3 phyla. Now, these 19 animal phyla are different from the 3 in the Precambrian. The Precambrian animals, the sponges and whatnot, are obviously extant today, so they're included in the Cambrian, but they're not part of the Cambrian explosion itself. We have 19 animal phyla, most of which are well skeletonized or mineralized. They've got exoskeletons or shells, which makes them easy to identify in the fossil record and much easier to preserve than, say, soft-bodied forms. But because of places like Chengjiang and places like Burgess Shale and some other places in Greenland and Newfoundland and the like, we have some soft-bodied representatives and are able to find these weird things like Eldoinioida, like the preapod and annelid worms, and cement their position in the Cambrian. So what happens after the Cambrian? How many more animal phyla are there in the first place, and what is their pattern of appearance? Not much. These are all of the animal phyla that do have fossil representatives in them, and it comes to a total of about 28 animal phyla, which means of the animal phyla that we have a fossil record of, 19 of 28 are first appearing in the Cambrian. This says nothing to speak of the 12 animal phyla of which we have 0 fossil record. Okay, so of a total number of animal phyla of 40, more than 1/4 of them have no fossil record whatsoever. They're very small, soft-bodied worms. Some of them live as parasites inside the guts of other animals. This is not going to be easy for a paleontologist to recognize, obviously, especially when we have very beautiful and delicate fossils. We don't want to muck with them trying to find a parasite that they may or may not have and may or may not be preserved. Some of them are little worms that live in what we call interstitial areas, or the regions in between sand grains on the beach. Beaches are not good places to get fossils. Okay, they're lousy, they're highly active, and so little soft-bodied forms are not going to be found there. And so what we have is a large number of the potential animal phyla in the fossil record being found in the Cambrian. Let's look at this a little bit better. Some of the animal phyla that appear after the Cambrian could potentially find themselves having a first appearance in the Cambrian. What I did was kind of a general qualitative study to look at the life habitats of these organisms, their hard parts, basically their preservation potential, irrespective of evolutionary arguments. We did not incorporate evolutionary arguments of, well, we think that these two organisms share a common ancestor somewhere in the Cambrian, so we're going to assume that the one that isn't in the Cambrian actually was. We're not making evolutionary assumptions here. This is purely based on a qualitative assessment of the fossil organisms as to whether or not, and the unfossilized organisms, as to whether or not they're likely to have their first appearance somewhere in the Cambrian, along with many of these other animal phyla. And it turns out that most of them could. Now, we don't know that they did, but most of them could, and we could have as much as 85% of all of the animal phyla making their first appearance in the Cambrian. This is astonishing. We go potentially from 3 Precambrian animals to nearly 35 animal phyla making a first appearance in the Cambrian. This is rather difficult for someone who is asserting a universal common ancestry due to descent with modification to account for. Now, Gould kind of understood this, stating that the Cambrian explosion occurred in a geological moment, and we have reason to think that all the major anatomical designs may have made their first evolutionary appearance at that time. Well, Gould's not entirely correct. We have animal phyla in the Precambrian. Indisputably, they are there. But he's getting close to the fact that at least potentially we could have as many as 85% of the animal phyla making their first appearance in the Cambrian, which is a whole boatload of little soft-bodied critters. So based on their first appearances alone, this is kind of the percentage that we have in the Cambrian. We have about 68% of the animals that have fossil representatives are first found in the Cambrian. Again, this is at the phyletic level. This is not to say that we have dinosaurs in the Cambrian. We have chordates in the Cambrian. We don't have shrimp in the Cambrian. We have arthropod crustaceans in the Cambrian. We're looking at these major morphological groups. I don't want you to misinterpret what I'm saying and thinking that every different type of form of life is in the Cambrian. No, but representative forms of all the major phyla are there. We have a few others straggling along in the Ordovician, which is directly after the Cambrian. The Devonian is stratigraphically and geologically later. Pennsylvanian, Triassic, and Tertiary, we have a couple fossils here and there. Most of them are just, we pick up one more animal phyla here and there, usually in very unique depositional settings. And so this is mostly considered, in many of these cases, a preservational issue, and not so much a problem with the idea that the fossils or that the organisms are actually first appearing in the Cambrian, but that we didn't have the appropriate conditions in the fossil record to actually maintain their position as a fossil. And so the ones that we have as fossil representatives that are found after the Cambrian are usually in very unique depositional settings that have high preservation potential, like the Cambrian does. So, for example, the subphylum of Arthropoda, we have the crustaceans, the trilobites, the anomalocarids, and several others. There are a few others. Very, very different. Shrimp-like crustaceans versus trilobites, which are named for the three lobes, that central axis that you can see behind the head, and the two side sections, those are the lobes of the term trilobite. And then the anomalocarids are radically different in their position. But at the subphyla level, we're starting to see a lot of disparate forms as well. So not only do we have arthropods, we got a few different major types of arthropods that may or may not be connected to one another. Well, we can do the same thing when we have all of the subphyla counted, and when we take a subphyla like arthropods, we drop out phylum Arthropoda and throw in the subphyla so that we're not counting arthropods an extra time. So we take all the different types of arthropods that have subphyla. We take the three subphyla of chordates, we have several subphyla of mollusks, and we redo the calculations and we take another look at the preservation potential of the organisms as to whether or not they can make a first appearance in the Cambrian. And overwhelmingly, at the subphylum level, which in some cases might arguably be just as important as a phylum level, we have 48 animal phyla and subphyla that could potentially have their first appearance in the Cambrian. That's a lot. That is huge. And again, we have a few unknown, we have a few extant living organisms with no fossil record, and we're not exactly sure where they might tie in. For example, a gut parasite of vertebrates, something that, like your dog has worms. Well, there aren't a large number of vertebrates in the Cambrian. There's a few, there's several, and there's several different types. But they're not a large number by any means, and they're certainly not mammals, and they're certainly not reptiles. And so anything that might be an obligate parasite to a mammal or reptile is obviously going to have a first appearance in the geologic record sometime later than the first chordate. And so these last five unknown groups that have extant but not fossil representatives, we're not exactly sure where to put them in the fossil record yet, but we do know that they're not in the Cambrian, so we don't want to overplay our hand by stating that they are when it seems clear morphologically and biophysically that they cannot be. But we look at this in comparison to what the Darwinian prediction is, and it's radically different. Darwinian evolution, be it neo-Darwinism or punctuated equilibrium, or any other theory that asserts a universal common ancestry based on this bottom-up version of evolution, is woefully inadequate and utterly wrong when it comes to the data of the Cambrian explosion. This is the high-end potential. Okay? The low-end potential is simply the fossil stuff, at least 19 animal phyla in the Cambrian. Either way, bottom-up theories are not accounting for the data in a way that a top-down view of evolution does, or a top-down view of design actually happens to look at things, which I think is more appropriate. And so, the basic summary of this particular section is that the fossilized phyla are coming in at a particular point in time, basically en masse. Sponges have little spicules that are kind of fossilizable, but they're certainly not like arthropod shells or mollusk shells that are very easily recognizable. In the Precambrian, we're not seeing those things. We're not seeing these fossilizable parts that, of anything, should have a greater potential for preservation deeper in the fossil record. They're not there. They're in the Cambrian, not in the Precambrian by and large. Again, aside from some of these potential forms that there's some bickering about in the geological literature. And so the fossils and the evidence are contradicting a bottom-up view of evolution that is predicted by all of the major forms of evolution, be it neo-Darwinism, punctuated equilibrium, or other bottom-up forms. So I'm going to flip back over to Steve Meyer as we start to deal with some of the possible objections to our position that design might be the appropriate hypothesis here.
Thanks a lot. In the article that's in this Michigan State volume, Marcus has summarized his research on this area in 3 appendices. And it's really very rigorous. I think one of the points that he was hitting on here that I'd just like to re-emphasize is that what he's done is a nice upper and lower bound analysis of how many new body plans come into existence in the Cambrian. And the reason that some of these possible forms that many paleontologists think some of the forms that have no fossil record start back in the Cambrian is that things like these gut parasites, you don't find them preserved, but you find the organisms, you find the phyla that have the organisms in which they reside first appearing in the Cambrian. So you deduce, oh well, if the thing is parasitic and its host first arises in the Cambrian, then it probably did too. So he's done a really neat analysis. He's not as sanguine as Gould about them all coming in, but he's not as pessimistic as just the raw data alone. And we've kind of, I think, hit the ball down the middle of the fairway and made a very responsible estimate. Either way, what you expect on the Darwinian model is that you have this gradual increase. You don't expect a sudden spike and then a few trickling in later. It just is not consistent. So on all of these fronts, then, neo-Darwinism and related theories have a problem with the fossil record. So what we've provided at this point is a critique of standard evolutionary models and their ability to explain the evidence of the fossil record as especially expressed in the Cambrian explosion. There's some significant objections to our critique of neo-Darwinism at this point that we want to at least touch on. We can come back to more in discussion. The first is one called the artifact hypothesis. And this basically says the Cambrian explosion is not real. It's an artifact of our incomplete sampling of the fossil record. And there are many different versions of this, but basically the idea is, one version is to say that we're not sampling the fossil record adequately, and there are differences in depositional environment that are responsible for the differences in the representation of fossils at different times. Now, there's some important research that's gone on at the University of Chicago with a statistical paleontologist named Michael Foote, and he's done a very, he argues that this is simply incorrect on the basis of sampling theory, and he's got a way of illustrating this. He says, imagine that you are blindfolded and you reach into a box and you pull out marbles, and initially you get greens and you get reds and you get yellows. You get the now primary colors, but you don't get anything else. If you've only reached in and gotten 6 or so marbles, you might be able to say, well, I have just an unrepresentative sample. But if you continue to sample the box and you keep pulling in and you sample thousands and thousands and thousands of marbles, and they all fall within these 3 disparate categories, at a certain point, statistically, you can no longer claim that your sample is unrepresentative. And he says this is what's going on in the fossil record, that on the basis of the breadth of sampling that we've been able to do and the fact that all new fossils fall within these existing taxonomic categories, or occasionally, which is what happened in China, some brand new taxonomic category, equally disparate from the others, is discovered. At some point you have to say, I think we're getting a representative sample, and this is what he concludes on the basis of his statistical analysis of the sampling of the fossil record. We have a representative sample, and therefore we can rely on patterns documented in the fossil record. Now there are a lot of different versions of the artifact hypothesis, and many of them have, they've all fallen by the wayside, but I want to get to we can talk about some of these, but there's one in particular, and the idea that the claim is that ancestors are seldom fossilized because they lack hard body parts. And it is true that soft body parts are more difficult to fossilize. But there's something that this objection doesn't reckon with, and that is that soft body parts of many Cambrian animals have been preserved. I think we showed in the paper that there were representatives of 15 phyla that are known from their soft body parts. And so it's simply not the case that soft-bodied parts are not preserved. And secondly, we have in the Cambrian we have many animals with soft-bodied parts preserved, and especially the Chengjiang is especially important because of so many soft-bodied animals and body parts have been preserved there. And secondly, there is now been an extraordinary discovery by some of the Chinese paleontologists, and our colleague Paul Chen has worked on this, and that is of Precambrian algae and sponges that are, the algae have been long known, but Paul Chen has been involved in the discovery of a sponge and the analysis of Precambrian sponges from the layers of strata just beneath the Cambrian strata in China. So you have the explosion, the Cambrian strata, and then the Precambrian strata there has been a discovery of sponges and even tiny sponge embryos in the earliest stages of cell division. And this is a micrograph here, and these are photos that Paul Chen has done in his work on this. And these little structures show the distinctive spicules of sponges, and so it's quite an extraordinary find. And here's the rub. If you can preserve a small microscopic sponge embryo in the Precambrian, then you ought to be able to preserve the animal forms that are alleged to be ancestral to all the other phyla from the Cambrian that arise in the strata just above these finds. Okay? If you can preserve an embryo, you ought to be able to preserve an animal. The transitional animals, the animals that you need to find to say that you have transitional intermediates and precursors to the Precambrian animals, are just not there. And for this reason, many of the Chinese paleontologists have really dismissed the artifact hypothesis, given in the back of their hand. I mean, we think this effectively kills it. There's a lot more to this whole discussion, but I wanted to brief you on these, I think, two key factors. And here's some more of Paul Chen's work. So I think that many of the leading paleontologists are very unconvinced by the idea that transitional intermediates, transitional alliances are present and we simply haven't found them. Here's Valentine and Irwin. They say transitional alliances are unknown or unconfirmed for any of the Cambrian phyla, and yet the Cambrian explosion near the beginning of the Cambrian time was real and produced numerous new body plans. So that's one of the objections you'll hear about most, the so-called artifact hypothesis. We just haven't found them. They were too soft, too small, but soft, small things have been found, and they don't do anything to fill in the pattern that the Darwinian theory requires as far as the tree of life. So we think that what we're looking at is something that's real and not an artifact of incomplete sampling. There's a second objection, and Marcus is going to deal with that briefly, and then I'm going to present our alternative, our theory of design, and how that explains these key features of the Cambrian explosion.
One of the other major objections that has come out against the Cambrian explosion is that in the Precambrian, we do have organisms that some have actually claimed to have been ancestral. And particularly, we're going to be dealing with what are called the Ediacaran fauna and what challenges they pose to the Cambrian explosion. The Ediacarans are a group of organisms that are known up to the Cambrian the Precambrian Cambrian boundary. And they're a highly bizarre group of organisms. They're not morphologically similar to later Cambrian animals. They tend to be these large, pillowy, kind of air mattress sort of things. They don't have eyes. They don't appear to have a digestive system with a mouth or an anus. And many people are fairly convinced that these actually are not, in fact, animals. And if you look at some of the geological literature about the description of these things, all over the map. They could be animals. They could be a sister group to animals and all other sorts of things. They could be an entirely different kingdom of their own. They could be a separate phyla. A lot of questions about the Ediacaran fauna. We do have animals in the Cambrian, as we mentioned. We showed the sponges, and that we have worms based on the trackways, and of course some jellyfish. And these things are in the Precambrian definitely, and they're in with some of these Vendian or Ediacaran organisms. Once again, our max and min phyla counts. Basically, the thing to understand about the Ediacarans, I'll just describe them briefly. Besides being these kind of large and high surface to volume area sort of things, one of the problems about dealing with the Ediacarans, and one of the reasons why descriptions of these things are all over the map in paleontology, depending on who you read, you'll get a lot of different opinions as to what they are and what their importance happens to be. Most of them are found in a rather coarse sandstone. The first of them were found in the Ediacara Hills of Australia. Since then, they've been found globally. They're all over the place. They're in Newfoundland, they're in Africa, they're on pretty much all the different continents, in the same geologic locations pretty much in terms of temporal statements, but they tend to be found in these very coarse sandy deposits. And what that does is that you end up decreasing your resolution on the organism when you have larger and larger sedimentary particles as your matrix. With the fine clays of Burgess and with Chengjiang, these things are the clay particles are microscopic, and thus are able to preserve intricate detail of the internal guts and the complex organs of these animals. That's lacking in the Ediacaran fauna. You've got sand particles, which are very grainy, and so you get basically a lot of imprints of these things and kind of maybe some sort of biological residue that these things were kind of pressed down, and then perhaps the biology of them or the soft parts all dissolved away, but they left an imprint on the strata, either above or below or both. They might move, they might not move. If they do move, it could be due to currents, it could be under their own power. There's a lot of questions about the Ediacarans that are very much unknown. And so if somebody's telling you that the Ediacarans solved the mystery of the Cambrian explosion by giving us precursors to Cambrian animals, well, that's one opinion, of which there are about 8,000, it appears, in the paleontology literature. So these things are a potential objection, but until we get any kind of resolution on the Ediacarans, which hasn't been forthcoming over the last 70 years, these things, in my opinion, appear to be radically different, and I think in the opinion of most paleontologists, are radically different from the animal phyla that we see later on in the Cambrian, and radically different from the animal phyla that we see contemporaneous with them in the Precambrian. They're obviously not sponges, they're obviously not jellyfish or worms. So we'll hit back over to Steve. Don't worry about clapping every time we come up here.
Sorry about that. You guys are great. This is the first time we've been able to do this together, so we talked about this at lunch over, so our baton handoff is not, we need another slide here, because we had a couple points. Marcus is quite right that there are certainly, many paleontologists doubt these are even animals now, and so there's a morphological disparity. They're not good candidates to be ancestral precursors.
Just to steal your thunder,
yeah.
Kind of the view that the Ediacarans are animals is an idea that was very popular back in the 40s, the 50s, and the 60s, when there were very few of these things known. They were still highly controversial, and people were kind of force-fitting the idea of animal phyla onto the Ediacarans. They had stuff that looked potentially, if you squint your eyes and close one, might look kind of like an ancestral trilobite. But as things have come out and we see these morphological forms perhaps in slightly better detail and certainly in more numbers, the idea that these things are actually animals is being held by less and less paleontologists, and those who do believe that they're animals are retiring because they're kind of old. So,
just a couple other problems with them. One, he mentioned there's a gap. They go extinct before the beginning of the explosion. Thirdly, the Ediacaran themselves represent an unexplained increase in biological information. They and the other forms in the Precambrian have definite origin points, and the same problem that you have in explaining the origin of the information in the new form in the Cambrian is actually replicated, though at a lesser degree, in the lower strata as well. So they don't really solve the problem of the Cambrian. They actually add another little bit to it, and that's kind of where I want to go now and talk about what we make of this. There's another objection called deep divergence. We're going to just simply let you know that there's another objection called deep divergence, and we can talk about that in Q&A if you like. What I want to do now is present an alternative model for understanding the origin of the animals, the origin of the animal phyla, the origin of the animal body plan, and this is the model or theory that we've been calling intelligent design. Most of us are familiar with some of the basic design theoretic concepts, that there are certain features that are better in the natural world that are better explained by intelligent causes rather than undirected natural causes. We have Mount Rushmore here on the right and the hills in South Dakota, I guess, on the left. Badlands, okay. And so there's a recognition in our thinking that there are some things that are explained by the result of intelligence and some things that are better explained by the result of undirected natural processes. What about the features of the Cambrian explosion? We've seen that the quantum increase in biological information, the persistence of morphological isolation, the top-down pattern of appearance, the sudden appearance without ancestry. Many of these features are things that are contrary to our expectations if we're holding a neo-Darwinian model. What does design have to say about these features of the Cambrian explosion? Could they be better explained within a design theoretic model? I'm going to start with the question of the origin of the biological information necessary to produce all this form, and show what design has to offer there as an explanation, and look at each of these other features that we've been discussing. I've slightly rearranged these for purposes of presentation. We talked about information as one of the last features of the Cambrian explosion. I'm going to talk about that first in our discussion of design. First, though, I want to make a methodological point about how we reason in the historical sciences. Because we're not present to see things happening, and because we can't always replicate past events in constrained experimental contexts, historical geologists and others, paleontologists, use a principle that is called uniformitarianism, or sometimes called actualism, probably more precisely. And the idea is that sometimes the slogan is the present is the key to the past. More precisely, the idea is that the cause and effect relationships that we see today in the present should guide our assessment of the plausibility of the hypotheses that we posit as explanations for events in the past. If you're a geologist and you're looking at mud cracks, and you happen to know something about the way that water recedes on lake beds and how they can get dried out and what kind of structures form, if you have the ability to sort of monitor an ongoing process and see what that process produces in terms of an artifact, then you can more readily reconstruct what produced that same artifact when you find it in a fossil strata in the past. This is a standard form of reasoning. Our understanding of cause and effect relationships now help us to understand what might have caused some effect that we're looking at in the past. Well, one of the effects that we're looking at, one of the features of the Cambrian explosion that needs to be explained, is what we call quantum increase in biological information, the non-continuous, discrete, big jump in the amount of information in the fossil record. What can explain that? Well, remember we talked about the need for more DNA. I came across this quotation, or this passage, in a early information theorist, pioneer, Henry Quasler. And Quasler was a pioneer in applying information theory to molecular biology. And he said, I made an offhand remark once, he said, the creation of new information is habitually associated with conscious activity. This is something that we know conscious agents do. They generate new information. And Richard Dawkins has noted that inside the cell, we have a machine code that is uncannily like the kinds of machine codes we see in a computer setting. He makes the comment, the machine code of the genes is uncannily computer-like. The pages of a molecular biology journal might be interchanged with those of a computer engineering journal. You put these two observations together, and I think you have something very suggestive. That is, first of all, that we know that there is information in biology. The information in, at least the genetic portion of the information, is digitally encoded. It's very like a computer code. And this kind of information we know is produced by intelligent agents. Intelligent agents are what philosophers of science call, they are causally adequate to produce this feature that we see in the history of life. So certainly intelligent design, on the basis of our uniformitarian knowledge, our knowledge of cause and effect relationships in the present, ought to be considered, or there's a reason to consider intelligent design as an explanation for the sudden increase in information, namely that we know that intelligent agents are causally adequate. They can produce this kind of a feature. And given that we know of no other feature that can produce this, we might consider design as the best explanation. Now let me elaborate on that latter claim, because we haven't really talked about the problem that the Cambrian explosion poses. Here's an illustration that intelligent agents can produce intelligent information. They can produce information. Now, we haven't really talked about the claim, the problem that the Cambrian explosion poses for the mechanism of neo-Darwinism, the natural selection random mutation mechanism. The key driving force in that mechanism is actually mutation. That's what generates the new information. is it plausible to think that mutations could have generated enough information to produce all these new Cambrian animals? Well, on a strictly quantitative basis, this is what's called the mutation rate problem, it turns out that it is not plausible to think this. Here's a passage from a scientific paper by Susumu Ono, who's commenting on this very problem. He says that even giving a very generous rate of mutation, it still gives, assuming an extremely generous rate of mutation, it still takes 10 million years to undergo 1% change in DNA base sequences. The emergence of nearly all the extant phyla of the kingdom Animalia within the time span of within 6 to 10 million years can't possibly be explained by mutational divergence of individual gene functions, assuming that rate of change. So if you give generous rates of mutation, you think they can only change about 1% of the DNA base sequences in existing organisms, you're just not going to get the kind of change, the amount of information you need to build these organisms. It's quantitatively out of the question. So there is just a problem of rate with mutation. There's also a problem with what we call the specificity problem. It's not just the amount of genetic shuffling, it's that genetic shuffling alone doesn't get you the specific changes you need. And this is really what's meant by information. This is a slightly humorous thing if you're reading here. It says, hey, I just mutated into a digestive enzyme. Cool, says the other piece of bacteria. Too bad you don't have an intestine. Okay, you don't have any digestive tract to use the enzyme. It's not just a matter of shuffling the genetic information. You have to shuffle it in such a way as to build new proteins and proteins that are actually functional for the organism that hosts you. Now, one way to get a handle on specificity is by analogy to language. If you think of repetitive sequences, A, B, A, B, A, B, they are very orderly, but they lack complexity. Complex sequences, like this gibberish here in the second line, lack what we call specificity. But things that are both complex and specific are things that we refer to as informational or having information content. And this is the kind of, when we're talking about the information that you have in biology, we're not just talking about improbability. If you're a math person, you know about Shannon information, which is a measure of merely improbability or information carrying capacity. Biology, the information in biology is not just Shannon information. It's not just improbable. It's not just complex. It's complex and specified. It's like language in that respect. And that's what has to be explained when we talk about the origin of the information necessary to build the Cambrian animals. We need long sections of code that are specific to the functional requirements of the organism. We don't just need a hunk of DNA turned upside down, rearranged, and shuffled. We need a hunk of DNA that has text that produces specific proteins. Now, there's some very interesting work that's been going on that is testing whether or not mutations could plausibly, in the best of cases, ever produce new proteins starting from existing proteins. In other words, there's experimental work testing the plausibility of the neo-Darwinian mechanism at the very bottom level, at the very base level. And one way to think of this is if you think of a gene as like a section of language, the question is whether you could mutate one part of the genetic text and maintain a continuous function as you find a new and radically different section of genetic text. Genes produce proteins. If you want to produce new proteins, you've got to morph the genes in such a way, you've got to change the genetic sequence in such a way that you continuously maintain function so that the genes are going to be preserved and the new proteins that they produce are going to be preserved. Well, there's a technique called site-directed mutagenesis that has effectively been testing how much can you change proteins, how much can proteins be morphed in their primary amino acid sequence as a result of genetic changes, how much can be changed and maintain continuous function and maintain function. Because in the Darwinian view, at the molecular level for evolution by natural selection to take place, you've got to have a series of at least islands bridging one major protein motif to another. You've got to maintain a continuous function. And the result of these mutagenesis experiments are showing that instead of having a series of little tiny stepping stones, or even better, a continuous pathway of function from one protein motif to another, the sequence space of proteins looks more like islands in a vast ocean. And there's going to be some very important papers coming out on this very soon. There's some things that have already been published that establish this disparity, and we discussed these at length in the Cambrian Explosion essay in the Michigan State book that we've been talking about. But this is the Darwinian picture at the molecular level, what must be so for the mechanism of mutation and selection to produce new information, new functional information that would result in proteins. And this is what protein sequence space actually looks like. And this, we think, poses a really fundamental objection to the idea that mutation and selection can build new specified biological information. And with that in mind, then, we're going to argue, we do argue in our article, that intelligent design provides a better explanation for the origin of one of the key features of the Cambrian explosion, namely the information necessary to build the new forms, and the information at the genetic level. So that's one aspect of our argument. A second aspect comes from the recognition, this was a point that Jonathan Wells and I were discussing across the way yesterday in the final session, is that DNA, as important as it is in building new proteins, and proteins are these incredible molecular machines and macromolecules, all DNA does is build proteins in the end. It does not arrange the proteins, at least not by itself does it arrange the proteins, into higher level structures. There are some proteins that self-assemble into some limited higher level structure, but when you talk about the architecture within the cell, when you talk about arranging cells into cell types and cell types into tissue types and tissues into organs and organs into body plans, much of this organizational work is done by epigenetic sources of information, some of which the sources of which are still somewhat mysterious. We know where some of that epigenetic information and patterning comes from. We don't know where it all comes from. But what we do know is that the information in DNA is not sufficient to arrange the small level components, the proteins, into these higher level structures. You might think of it by analogy to a computer where you have some instructions that help you build electrical components, some manufacturing instructions, resistors, capacitors, the like, but then they have to be arranged into a circuit board, and the circuit board has to in turn be arranged into the larger computer. The computer could be in turn arranged into a network of computers. The information for building the components does not determine that higher level organization. And so I made the point yesterday in the discussion with Jonathan that what this means from the standpoint of for neo-Darwinism as a mechanism for generating the kind of information you need to build a body plan, the mechanism is, I think, effectively dead. And I think this is going to be increasingly recognized. It means that you can mutate DNA indefinitely without respect to probabilistic limits. That is, this is not a probability problem anymore. It's not a question of, well, would we get lucky? All that you could get in the best of cases mutating DNA is if you are extremely lucky, you will get a new protein. We think that's implausible for the reasons I've already stated. But you're not going to get this hierarchy of form and information that a new body plan represents. If DNA only builds proteins and you need other sources of information to arrange proteins and other parts into body plans, then DNA alone, mutations in DNA alone, which is what neo-Darwinism asserts, is not going to be sufficient for body plan morphogenesis. So we think this is a fundamental in-principle argument against the sufficiency of neo-Darwinism as an explanation for the higher level forms of information you need to build new animals. Now, with that in mind, if we think about design, and we've already made an analogy to the way in which design systems are hierarchically organized. Design systems are made of information-rich components, but those information-rich components are in turn arranged in highly specified, that is to say, information-rich ways. So we have a hierarchical organization of information. You have information packets within informational structures. That's what designed, that is a feature of design systems. That is also a feature of organisms, of animals, and in particular the Cambrian animals. Where you could say that hierarchically organized information is a hallmark of intelligently designed systems. Certainly we know that the neo-Darwinian mechanism is not, I think now very definitively we can assert and argue that the neo-Darwinian mechanism is not sufficient to produce that kind of a feature. We know of no other causal entity from our experience that can produce information and information organized into these kinds of hierarchies other than intelligent engineers. And so when we look at this feature in the history of life, we can, using our uniformitarian logic and principles, infer design, intelligent design, as the best explanation, based on what we know from our present experience about the cause and effect relationships that exist in the universe. Mind is the only cause that's sufficient to produce information and information-rich hierarchies, and yet that is one of the key features that needs to be explained in the Cambrian explosion. So here's our, in a sense, we can get at this very intuitively by looking at this kind of I have a friend who has given us a slogan, technology recapitulates phylogeny. Okay, where the Greeks used to say, what was it, nature imitates art, or art imitates nature, art imitates nature. And so we're saying maybe this ought to tell us something about the origin of systems that have these features. Now let me talk about a couple of the other features of the Cambrian explosion a little more briefly, and then we'll get into some discussion. Another feature that needs to be explained is this persistent morphological isolation of the body plans. It turns out, and we've described this already, so I'm going to press on, it turns out that this also is a distinctive and unique feature of humanly designed systems. I hinted at this when I first discussed this, but our own high-tech world is characterized by not by organisms, but by technological systems that are morphologically disparate from each other and which lack intermediate connecting things, because the connecting technological objects, the morphed versions, if you tried to make a plane and an airplane together, are typically non-functional. And so this persistent morphological isolation is a feature of intelligently designed systems. What philosophers of science would say is that intelligent design is, this is the term, causally adequate. You judge a theory as to whether or not it provides a good explanation by judging whether the entity you posit in the theory is known to be able to produce the effect in question. Intelligent agents are known to be able to produce morphological isolation. That is a feature of design systems, a hallmark of design. And guess what? We find that in the fossil record, and it is a particularly striking feature of the Cambrian fossil record as well. So we think of a kind of comparison here of the effect, and we know that the cause in one case of this type of effect is intelligence, and absent a better causal explanation for morphological isolation, certainly you don't expect this on neo-Darwinism, design is again a better explanation. Now let's look further at this related feature of the top-down pattern of appearance. We've talked about this. You get the big differences first, and then the diversity within the big differences later. This, again, happens to be a feature of our own history of human technology. Somebody talked yesterday, I think it was Jonathan Wells, talked about the Triz people yesterday. There's an interesting story about Niles Eldredge, the famous paleontologist who does work on punctuated equilibrium. He loves to study the, quote, evolution of the cornet, the trumpet-like instrument. And he sees all this variation on primal themes, and he describes this as evolution. But it's top-down evolution, and of course we know in the case of the coronet that it is not undirected top-down evolution, it is designed. One of the features of design systems is precisely this having an original design innovation that can be represented as a bowel plan, a way of structuring and ordering parts to perform a function, and then subsequent innovation on that basic primal technological theme. We see this with automobiles. All automobiles since the inception have had this basic 4 wheels, 2 axles, drive shaft, certain basic functions in common that define the body plan of an automobile. But now we have front I-beam suspension and stereo surround sound and all kinds of different variations and themes, variations on that basic theme. This is a feature of our own technological innovation, is this top-down pattern. And yet, again, we find that in the fossil record. So we argue that intelligent design provides a better explanation of that because intelligent designers are known to be capable of producing that effect, and the Darwinian mechanism and other similar mechanisms are not. And finally, even the appearance of sudden, even the idea of the feature of sudden appearance, I think is better explained by intelligent design. People are a little nervous and titchy about invoking design to say, poof, there's something suddenly emerged. But there's a way to think about this that I think gets us in touch with something profound. If you're going to explain the origin of the animals from not just an evolutionary point of view, but from any materialistic point of view, a non-design perspective that excludes mind and relies wholly on matter and energy, you will necessarily have to do it from a bottom-up approach, where the material parts have to precede the existence of the form as a whole. There must be a material antecedent on a materialistic theory of the origin of the animals. One striking feature of the history of life and of the Cambrian explosion is the complete absence of those material antecedents. So not only neo-Darwinism, but really any materialistic evolutionary theory requires evidence of those material antecedents, of the parts prior to the whole. Now, the only way you could get around that is if you had a compelling artifact hypothesis, and I think that is no longer the case. So now think of the design explanation. How is it that there are no material parts before you get the material whole, before you get the body plan as a whole? Well, this is actually how designers work. They do not instantiate the matter first. They start with an idea and then arrange the matter in accord with the design plan. So the absence of transitional intermediates is contrary to Darwinism, and Darwin actually stated this. He says, natura non facit saltum, or non facit saltum, depending on how you pronounce your Latin. Nature takes no sudden leaps. Nature does not have that causal capacity based on our knowledge of cause and effect relationships, using our uniformitarian reasoning. On the other hand, we know that minds can leap to new designs. We know that an idea often precedes, almost always precedes, a new design innovation. And so the absence of transitional intermediates is consistent with a design explanation, but utterly contradictory to the neo-Darwinian mechanism unless they have a compelling artifact hypothesis, and that's no longer the case. So what we've actually looked at, we think, are there are four features of the Cambrian explosion that are extraordinary and perplexing from a neo-Darwinian and or other materialistic evolutionary perspectives, and yet are known to be features of design systems. And for this reason, we make what we think is a very strong cumulative case, based on the presence of these features, that design is the best explanation. And one way to understand the method of reasoning here, we are applying the standard method of reasoning used in the geological and paleontological sciences. It's called the method of multiple competing hypotheses. And the idea is that you compare many competing hypotheses as to their explanatory power. This is the way you test ideas in the historical sciences, by the way. We've been talking a lot about predictions. In the historical sciences, you test hypotheses against their competitors to see which has the best explanatory power. And if you test neo-Darwinism, punctuated equilibrium, and intelligent design against the data of the Cambrian explosion, we think that only intelligent design can explain the key features of the Cambrian explosion, and neo-Darwinism, Punkic, and other similar undirected evolutionary theories lack the ability to explain one or more of the key distinguishing features of the explosion. And for this reason, we're in a sense resuscitating the great argument of Louis Agassiz, who was the leading paleontologist of the 19th century, who dissented from Darwinism till his death, not on religious grounds, but on the basis of the paleontological evidence. And Agassiz argued that, in particular, the sudden appearance of the fossils in the Cambrian, he said these tell of acts of mind. He thought that this was evidence for an intelligent cause, what he called an intervention of an intellectual power. This is what he thought the Cambrian explosion attested to, and he never accepted Darwin's case. Darwin, however, at the time argued that the fossil evidence was simply missing or misleading, and so it since came down at that time to the argument between design and the artifact hypothesis. And Darwin held this in part because he felt that there must be a naturalistic explanation. We must have, to be scientists, we must limit ourselves to naturalistic explanations. We can't consider design. And what we want to do is, in a sense, reframe that question and say, let's let the evidence speak. And rather than limiting ourselves to the best naturalistic explanation, we side with Agassiz in saying that we should be seeking the best explanation of phenomena in nature, even if it includes something, even if it includes design. So if we map the cognitive terrain here, we have three possible explanations. If you hold to the convention of methodological naturalism, you have to exclude design by definition. And we're simply not willing to do that because we think as scientists, it's important to have the theoretical freedom to infer to the best explanation, not just the best explanation among an artificially limited set of options. And so we want to remove that boundary, and once you do that, we think the evidence speaks strongly in favor of design as the best explanation for the key features of the Cambrian explosion. And we'd like to take your questions. Yeah, we got one right. Oh, somebody right here at the mic, yeah.
Yeah, I'd like everybody to come up to mics. Thank you very much for both of you for an outstanding presentation. It's obvious that we've been standing too close to the tree of life to see the forest of life.
Good. Thank you, Art, as well, because Art wrote a very seminal essay on this back in the early 90s. And some of this work really builds on those seminal insights, and we're indebted to you. So.
Well, if there was a little bit of a
so maybe that'll make your question nicer to us.
You've sure built a wonderful building on top
oh, good. No, that's a great paper you did. Yeah.
Thank you. I'd like you to comment on the differences between a weak ID theory and a strong ID theory. Now, a weak ID theory would basically be to show that naturalistic explanations are insufficient to account for evolution, hence you infer ID. An example would be the diversity of species in the Cambrian explosion is low. Hence, Darwinian processes have not had a very long chance to act. Therefore, Darwinian processes did not cause the origin of the phyla, and these new phyla have not been on the scene very long. Even if you erased the entire geologic record below that, the diversity is still low based upon the sampling. Okay.
Yeah, that's another way of saying, for the audience who are not paleo geeks, that's another way of saying the top-down pattern or disparity precedes diversity.
Right.
Yeah.
But
right.
even if you
something unexpected on the Darwinian.
Even if you were to erase everything and all that fossil record is missing below that, the diversity is still low.
Yeah. Right.
Okay. At that point. Okay. The strong ID theory would be explaining the natural processes that inhibit major evolutionary change from occurring. All right. You mentioned stasis, you mentioned conservation, you showed examples of the phyla persisting through long periods of time. It would seem to me that a strong ID theory could be developed using even Darwin's own mechanism of natural selection, which would explain maybe, the elimination of useless transitional stages like your caroplane or your air car. That would probably take an airhead to own one in the first place. Anyway, in developing a theory for natural processes that prevent major evolutionary change from occurring. I'm just wondering.
Well, that's excellent. And of course, Art, you've kind of shown the way in some of this by talking, about how natural processes can give an explanation for stasis. Let's go further. I'm going to sketch this out a little bit. Weak, strong, and stronger. Okay, because what we're presenting here is not only strong on your terms; it's stronger than that even. And here's what I want to unpack that. Many times, people will say that the design argument is an argument from ignorance. Let me first do your point. Okay, there's a reason for persistent morphological isolation, and that's somewhat implicit in this, even in this presentation. There are several reasons. One is you can't get proteins to morph enough. These new studies in site-directed immunogenesis are showing you can't get proteins to morph enough to produce macroevolutionary change by natural selection. That multiple position changes in the primary amino acid sequence inevitably lead to a loss of function before they ever help you find new function out in that vast combinatorial space of possible amino acid arrangements. That's what's coming out of the molecular biology analyses. That's one reason for stasis, why you can't change very much. Another has to do with embryological mechanisms that, that simply either destroy or shut down development if you start mutating things. In the paper, we deal with this extensively, that the kinds of mutations you need to produce major morphological change have to occur early in body plan development, yet those are the ones that are invariably lethal or deleterious. The kinds you don't need, ones that occur late in development that affect only somatic cells, are great. They produce minor changes, but they don't produce new body plans. So there are mechanisms, both embryological and molecular, and these are only two that I mentioned, that I think constrain the amount of variability you can get within the overall typology or picture of the history of life. So I think this also has some empirical implications for the discussion yesterday about monophyly, or, is intelligent design open to the idea of the universal tree of life, the idea of universal common descent? I would say yes, it's open to it on in principle grounds, but I think the evidence that we're looking at from the fossil record and from embryology and molecular biology say that what we have is a profoundly discontinuous typology for the history of life. And I think that as intelligent design, the research program, develops, our ability to sketch in those details is going to help us answer another objection, which is, hey, you guys don't have any grand narrative like evolution offers. Well, we're starting to sketch that in. As you see, it's not just evidence for design at the point of the origin of life or at the point of anthropic fine-tuning. Now we're seeing evidence for design at a particular point in the history of life, and with that, a more detailed understanding of the typology of the history of life. Now, the final thing, that's strong, okay? That's stronger than just Darwinism can't do it. We're also saying why Darwinism or other mechanisms couldn't fill in the gaps.
Well, you're also explaining natural processes.
Right, how natural processes are at work in that.
How they actually work.
But we're doing one other thing that I think is very important. And I want to be very self-conscious and have people see the method that we're using here. The other claim that's made against design is that we're making arguments from ignorance. Darwinism can't do it, therefore it was designed. That's not what we're doing. The logic of this argument is it's not an argument from ignorance. It is an inference to the best explanation. We have several competitor hypotheses, and we are assessing which of those hypotheses, which of those entities can best explain the evidence that we see based on our present knowledge of the cause and effect relationships that obtain in the world. And it is part of our knowledge base, not part of our ignorance. It's part of our knowledge base that intelligent agents can produce these four key features of the Cambrian explosion. So this is not just an argument against Darwinism or an argument from ignorance. This is an argument from our knowledge of the cause and effect structure of the world and the features of life as to what would best explain life. It's provisional and scientific, but it's the same, it turns out it's the same form of argument that Darwin used in The Origin of Species in making the case for universal common descent, and it's the same form of argument that's used in the geological sciences. So if you fault this as an argument from ignorance simply because things might change in the future, you have to fault all of science. I mean, this is a standard scientific form of reasoning, and it's not what philosophers term an argument from ignorance.
Thank you both again. Outstanding.
We're out of time. I'm sorry. What are we next? Going to lunch? We're happy to hang out longer because the next thing is lunch. So if anyone else wants to ask questions, go right ahead. Yeah.
Okay. I very much enjoyed your presentation. I especially found the molecular evidence that you hinted at, or at least said about, if that really pans out, quite convincing. What wasn't clear to me is why neo-Darwinism would require a bottom-up approach instead of the top-down approach, because it seems like, just like as you mentioned in the case of intelligently guided evolution, when you have evolution of cars and things like that, or the evolution of a computer code, that early on in the development, let's say, of computer codes, operating systems, you have a lot of room for major conceptual innovations, the kind of basic core structure in which you're going to, around which you're going to develop the code. But then later on, once you say you've got the Microsoft Windows developed, you only can do minor variations now.
Right.
And so your major kind of large-scale plans are, the core concepts are fixed, and that's analogous to the phyla. The phyla are the big ideas, the core concepts. So it seems like, for just that reason alone, early on, the major body plans would be fixed, and so then you would expect only variation from them. And also for the developmental reasons you talked about, if the early body plans are what are determined early on in the developmental stages, then you would not expect them to be modified as evolution occurred, because that would destroy the organism. So how would you respond to that sort of objection to the claim about what you would predict under neo-Darwinism?
Well, I'd say that everything you say is true, but it's not an objection, because the thing that's presupposed in that is the origin of that source code in the first place, and that's the big problem. Stuart Kaufman makes the same point. He says, yeah, we would expect, once you have the body plan, that constrains the amount of variability, but there's a lot of unoccupied niches, but he never explains the origin of the body plan itself. Once you have the body plan, yes, you can get diversification within that, but the body plan takes a lot of information, both genetic and epigenetic, and in the Darwinian approaches, you have to get that bit by bit as you get the small mutational changes. The bottom-up approach is necessitated, A, by the fact that it's materialistic, and B, by the fact that it's gradualistic. You've got to assemble the whole from the parts first, and the parts have to be built at the very baseline, or at the very basic level, by changes in the genome.
Well, it seemed like you presented two separate arguments. One argument was this information argument, okay?
Yeah.
And then you presented a second argument that neo-Darwinism predicts that, and that was the graph up there, that you would, instead of having all the phyla occur near the beginning and only a few come later, that you would expect this gradual increase in phyla.
Yeah, and
it seems like that prediction
no, it's essential to it, Robin, because it's the nature of the Darwinian mechanism itself that requires the bottom-up approach. There is no, you don't do, there's no macro mutations allowed, and Kaufman is now wanting with his self-organizational approach. He's trying to, he recognizes that, so he's invoking what he calls long jump mutations to explain the origin of the Cambrian. You need a big jump. It's still bottom up, but it's bottom up in a great big jump. The problem is that Kaufman acknowledges that the kinds of mutations that affect he actually acknowledges what I was talking about in embryology, that large-scale mutations early in development are inevitably deleterious. So in order to get around the problem, he has to invoke the most implausible form of mutation. And in a sense, both of these evolutionary approaches are in trouble. We develop this in a critique both of Punkgeek and self-organization in that same paper. But
okay.
it derives from the nature of the Darwinian mechanism. You've got to build it little by little as a result of genetic changes. Those affect proteins. Proteins are little tiny parts. The Darwinian approach is to build the whole from the tiny parts. That is bottom up. Okay.
Yes, I have a question about ecology. Maybe you mentioned this and I missed it, but I noticed that there was the mention of parasites, and also with the Anomalocarida, that they had these specialized appendages for feeding upon trilobites. And this seemed to be fairly well documented, which to me indicates a complex predator-prey relationship, which to me would seem to be another design argument. Could you comment on that, please?
Well, that's a tough one. We haven't exactly gotten into the idea of design within ecosystems in the paper, neither in the paper nor I think has really many people in ID kind of ventured out there.
Forrest Mims is doing a huge amount of work in this area.
Okay, so Forrest Mims is working on this. The idea of a well-balanced ecosystem has always been something that has been intuitively appealing to both design proponents and also to Darwinian evolutionists. I mean, both of them think that you can get a well-structured, functional ecological system, and we invoke different mechanisms in order to do this. I mean, one way to do it is to impose laws in a way. Kind of, if you want to take for a weird analogy, Adam Smith's Money World, or Adam Smith, not the show, but Adam Smith. He wanted to get a well-ordered economy, and he said there were two different ways. You can get all the smart people who understand how to make money and how to run an economy into a room, have them argue and whatnot, and come up with a series of rules and laws, and then impose that upon the people, and this is how you get a well-ordered economy. Or the opposite potential is that you loose all the laws, get rid of everything, and allow competition to run for itself. So in a way, actually, in Darwinian evolution, you've got not only Malthus, but in a way, Adam Smith, because you've got this ruthless competition, and out of that emerges a complex, coherent ecosystem. So whether or not ID is going to be able to bring to the table, a concept of perhaps external laws, or even, depending on one's bent in ID, internal laws as to what ecology makes, is going to be a good question. I think that it's one that we can probably address. I'm not sure when.
I was just told, this is anecdotal, but in a conversation at this conference, that somebody who's doing work in ecology, that a lot of ecologists are getting really dissatisfied with the implicit use of design and teleological kinds of concepts in describing these things, because there is often an irreducible complexity to the overall ecosystem balance. So certainly Lynn Margulis and her kind of interesting approach might be, some of her insights I think might be interconvertible over into a design framework. But it's a great area for future work. Yes, sir.
You mentioned the emerging views that, or realization that to make a body requires information outside of just the DNA to make proteins. Are there speculations about other sources of variation besides DNA mutations that might help rescue the story?
You mean for the neo-Darwinists?
Well, I guess if it were non-genetic variation, I'm not sure you would use the word neo-Darwinist.
Yeah, that's right. It would be a new form of evolutionary
Is
theory.
there some other mechanism of variation that's being speculated?
That's a very good question. We do know, by the way, where some of that other information is, and we discuss a little bit of this in the paper. One way to illustrate this is like the cytoskeleton is made of identical protein subunits, okay? But the way those are arranged determines the structure of the cell. So the DNA makes the subunits, but it doesn't determine how to arrange those subunits, the building blocks in the cytoskeleton. And there are features of the cell membrane that help determine those arrangements. So that's where there's a kind of patterning. There's also information in the patterning of the maternal egg. But and I was first alerted to this in talking to Jonathan Wells back in 1993. And one thing that he pointed out was that as you get to these information not encoded in these molecules, but rather in the structure of larger, more macroscopic cellular entities, you're dealing with things that aren't going to be amenable to mutation because of a little hunk of radiation hitting it or something. The larger these things are, the less likely they are going to submit to the less variability that you're going to be able to get. And I think even more importantly, and this is something we deal with in the paper, is that the kinds of large-scale changes you need in the structure of embryos are things that are going to be inherently deleterious. And so we know that a lot of this epigenetic information is necessary to determine embryological development, and that the changes in development, you alter development early and you destroy organisms. So I would be if I were on the naturalistic evolutionary side of this argument, if I really were coming to grips with this, I think I'd be very pessimistic about finding other sources of variability, because you're right, in classical Darwinism you have natural selection and you have a source of variability. Neo-Darwinism said it was genetic. Question, is it possible we will find epigenetic sources of information that could rescue the basic selection variation idea? I'm deeply skeptical about that, and because the epigenetic sources are much larger and less susceptible to variation without deleterious consequences for development.